Hi, everybody. Welcome to the Senior Doctoral Fellowship last presentation of our wonderful Senior Doctoral Fellows research. Today, we have two excellent presentations from two wonderful fellows who have been getting to know over the year. We have Lu with us from human biology. We have Amber here with us from Buddhism, psychology, and mental health. Both of them are working on really, really interesting projects, and we're delighted to have them both here to share their research and to engage in conversation with us. We are going to begin with Lu from human biology, and at this point, I'm going to turn it over to the program director for the Senior Doctoral Fellowship project in human biology, my great colleague, Colleen Dockstetter.
Thank you so much, Tara. So I am very pleased to introduce YiQing Lu. I say Lu. Lu, can we all call you Lu?
Oh, yeah, sure.
Okay.
Hello.
He's known affectionately to us as Lu in the HMB program. He's doing his PhD in molecular genetics at U of T, and right now he's at Mount Sinai looking at novel regulators of wild type and mutant P3 stability, which is important when you're researching cancer, and that's what you're going to talk about today too, right, Lu?
Yes, indeed.
Lu has been extremely busy, not only with his research lately, but he's been doing actually quite a bit of teaching in the HMB. So, of course, he's the academic fellow here, as you know. He's also been an instructor with the Department of Microbiology. He's been teaching math at the Study Academy outside of campus, and he was the course coordinator lecturer, and in the past, the head TA for our genetics labs, HMB 311, and our health and disease labs, and a whole bunch of things. And Lu taught our labs for the first time in September?
Yes.
Or in this semester?
Yeah. Last semester.
And last semester, taking over Dr. Diaz's place on sabbatical. So Lu was thrown into the fire this year, into the fire. He's done everything now. So I look forward to his research talk because it's what I actually know the least. I know him as running around helping us teach as much as possible this year. So here's Lu.
Okay, great. Thank you so much for the kind introduction. I guess I should update my CVs a little based on this. Then thank you very much for this, and also thank you, Tara. Let me see if I can see the slides. Let's just give me a thumbs up if I can see if everyone can see my slides.
Good,
Cool.
Lu.
Thank you very much. So yeah, I would like to begin by thank the new college community, and also thank you, Tara, Colleen, Nancy, and everyone for this great opportunity and this scholarships, and also the great opportunity to host so many exciting activities in my role as a senior doctoral fellow this academic year. So I guess today I want to present a small part of a project that is taking place during the tenure of the scholarships. So let me just see this. Yes, so it's titled as Genome-wide CRISPR screens identify novel regulators of wild type and mutant P3 stability. So it's a mouthful of words, but we'll dissect this in a very layman manner. So the plan for our discussion today is as follows. So I'll begin the introduction with a story, and then we'll move on to the actual meat of the project, which is the screen itself, and also the follow-up, the biological functional follow-up of what we have been doing. So I guess the biggest question is that why should I care about P3 stabilities, and why should I even care about cancers? So I just want to throw in scary words. So cancer is ubiquitous; it's everywhere. We can get cancer anytime, everywhere, and fortunately, there's no escape. If we do not die of cancer, probably will die with it. And so thereby, cancer research is not only about passion, but also of everyone's, I guess, responsibilities. And of which cancer biologists will or anybody actually are close to even to cancer studies will probably hear about this protein called p53s, which is I would say the guardian of the genome that guarded our bodies from actually getting cancers. So I'd like to actually begin my entire discussions by actually a quick story of the discovery of p53s. So as many of our folks in the audience will actually start their summer research very soon. So I want to actually throw in the story to actually shed some light as to what we are about to expect in the summer to come. So first of all, just as we are right now getting crazy about the coronavirus and everybody is chasing the coronavirus, just about 50 years ago, roughly 50 years ago, there's another type of virus, which is called a tumor viruses. And that actually it's a virus that can actually causes tumors. And people want to find out why the virus want to cause tumors. And so PP3 actually surfaced at around that time. So it turns out that PP3 is interacting with the viral oncoproteins. And PP3 indeed is found in all of those tumors that are induced by viruses, and they are present in high levels, found in high levels in those tumors. And very shortly, scientists rushed to actually clone the cDNAs of those PP3, so-called PP3s they actually studied. They actually found out. And then by observing, by overexpressing those cDNAs, they were able to see that the cells are being transformed. In other words, they are being turned into cancers. Same time, though, there is a silent minority of people who actually made also very different observations, or I'd say completely different observation, or the opposite observations. In other words, their cDNA cloned were unable to actually transform the cells, but actually they will stop the transformation of the cells. However, given the status quo, where actually the mainstream kind of hypothesis around that time, those people actually silenced out, didn't really say much about it. So given the strong evidence just now I have just mentioned, such a strong association, so it's very natural to conclude PP3 is an oncogene because it kind of interacts with the tumor-causing viruses. But wait a minute, this is not really what we have heard about in HMB 265, where we learned PP3 is actually a tumor suppressor. So what is going on here? So it's not until 10 years later, well, or short 10 years from then, people then actually realized what the wild type PP3 sequences actually is. So it turns out that like most of the sequences or the cDNAs actually people actually cloned for were used back to the early 80s were actually mutants. And so thereby those PP3 mutants are oncogenic, but yet they are mutants. So they are not the true PP3 that are being studied. So short story, what can we actually learn from it? So the first message is that association, however strong, does not mean causation, but also how do we interpret a result? And it really depends on the context. And what's even more important is that we can change our hypothesis, but do not change our result, and have faith in the observations and document everything. And in our summer research, there would be a lot of hypotheses established, but how do we interpret it? And this is up to us. And so as a science student, I would say that we should really be a good scientist and document everything. And this will be very helpful to for future research. But as a side, in the scientific manner, though, we actually know that there also exist those mutant PP3s, which are actually oncogenic. So as it turns out that PP3 is actually, as we all know now, PP3 is the most commonly mutated genes in all cancers. More than 80% of the cancers actually harbor some kind of mutations in PP3 or its associated pathways. While most of the PP3's mutations actually will cause the loss of the tumor suppressive functions, more than 50% of those mutations actually occurs in a few very specific regions. So let's call them the hotspot mutations. Those are indeed the ones that are actually being studied back in the 80s. So as we mentioned, those mutations not only lost the tumor suppressive function, but they actually could function in a dominant negative manners, gaining the gaining the oncogenic functions. So I just want to pause for a quick second if there's any questions for about the PP3s or discoveries or this concept of a gain function gain function PP3s. I guess in a nutshell, I just want to say that it is just one single base pair of mutation of this entire 400 amino acids proteins. It turns a would-be good policeman into a bad gangster. So just one amino acid change. I guess there is no questions, and I will move on to our real discussions then. Yeah. So the question is then, how does those genes actually gain function mutations PP3 actually causes cancer then? So on our first glance, we actually turn into our mouse models. So in this mouse model shown on the left, is that this mouse actually carry a germline mutation in this gain function mutations. As you can see that in the tumors, this is an immunohistochemistry plot for PP3s. As we already know from the 80s, those tumors are have a strong stain for PP3s, in this case, mutant PP3s. What's interesting though, is in the surrounding stroma or the normal tissues, the PP3 stains are very low. So this already gives us an initial hint that a strong PP3 stain is levels are likely associated with a tumor formation or metastasis of formation. Interestingly, if we manage to actually conditionally cut out the expression of those mutant PP3s, like shown here, and we were then able to actually cause the tumor to collapse. In other words, if we were to actually remove those stabilized mutant PP3s, then there is a chance the tumor will actually be gone. So I guess the billion-dollar question is then what actually causes this stabilization of mutant PP3 in the first place? And if we know this, there is a really good chance we can stop this error to pushing downwards, instead pushing it outwards, and thereby as a cure for patients that are carrying those mutations. So as we know, in wild type cells or in normal cells that are not cancerous, PP3 levels is actually being kept at very low, mostly through a E3 ubiquitin ligase called MDM2. In such, MDM2 tagged PP3 for ubiquitin proteasomal degradations. As such, in those cells, the PP3 levels are low. Somehow, this happens in the mutant. We don't know some random force that actually causes the mutant PP3 to be stabilized. And what is this actually random force? We don't really know. And that's where in the 21st century, I guess, with the advent of the genome technologies, now we are able to actually, we are equipped with arsenals to actually find out what are the genes or the factors that are actually affecting this process. And thereby, my job or our goal hypothesis is to actually use genome-wide screens to actually find out what are the genes that are actually regulating the steps causing the PP3 to be stabilized. So this is the question we want to figure out, and how do we do that then? So again, back to the HMB265 courses, we know that if I want to show that a gene actually causes a phenotype, a B, and best way would be actually, what if I just remove this gene from the cells? Then this phenotype should not be there anymore. And then as such, I would know that gene A is somehow causing this phenotype. So if I do this actually for every genes in the genome, there's anyway 25,000 genes in our bodies. And if I do this for the entire 25,000 and pin them one by one, and then we would probably solve this problem. And thanks to this very technology called CRISPR, which kind of started to surface after 2013 or so, and we were now able to actually achieve this goal very easily. So just in a nutshell, what does CRISPR do? And first, this is the CRISPR complex, originally a bacterial host defense systems, and this has been engineered by humans, but by scientists to actually causes genome editings. So in brief, it has some really core component, which is the Cas9 enzyme complex. But also I want to show you is actually this CRISPR guide sequence, which will be specifically targeting our desired region. And so if I want to actually target the entire genomes or every gene that I want to actually study, all I need to do is to actually design different guides that are actually targeting each of the gene sequence. So downstream of this event, then the Cas9 will actually causes a double-strand break on the genome, and the cells are screaming to repair them. So in a rush, the cells will, the repair mechanisms will inevitably introduce some kind of mutations. In this case, the frame shift in this case. And as we know that once the frame shift is being introduced, then the gene functions will be lost in this case. In so doing, CRISPR allows us to perform something called a CRISPR loss of function screens. So this becomes the premise of the rest of our discussion. So in terms of in our purposes, so we have the tools, which is to use CRISPR to study these genes. Then the question is that how do we know if p53 is stabilized or not? So in order to study something, we need a really good assays. And our assay in this case is to use a reporter to study the p53 stabilities. In this case, just in a really brief introduction, that I fuse p53 with a GFP proteins. In such, I can actually approximate the p53 levels by the GFP intensities. But how do I know if this is actually affecting the global gene expression or is actually affecting affecting the post-translational modification? The way to do it is by introducing a internal controls. In this case, is the 2A peptide linked by a RFP. So in other words, if translated as a single proteins, but cleaved off as two separate entities, subjecting to different post-translational modifications, if this were actually indeed due to translational controls, we would actually observe this pathway 1 in this flow cytometers readout, whereas both the p53 GFP fusions and also the RFP proteins will see elevated signals. If, however, this is actually due to post-translational modifications, then this is, the RP level will stay the same, and only the ones that are genes are only actually regulating the p53 GFP fusions. In this case, which is this step number 2, this will be actually of our focus. In this case, we're going to bring this machinery into a cell line that has been engineered to express Cas9 to facilitate the screenings. And this will be the rest, will be the workhorse for the rest of our study. Just in a even more brief introduction about how the CRISPR works is that, as we mentioned, we create a library that is actually targeting every single genes in the genome. And then I bring them into the cells, whereas I can make sure that each cell will receive one guide at a time. And so that each cell has only one gene being edited. And then I can then assay each individual cells and to see that what is the effect on the p53 stability of each cells. And then I can then study those cells to see what is actually being found in those genes, inside of those cells, and what the guide is. And then I will know that what gene is actually being edited in order to actually affect the p53 stability. So as we know that I just mentioned that MDM2 is actually a guide that is a gene that can actually, whose loss will cause this p53 to elevate. In this case, we can see that if I knock out MDM2 guides, it will cause its p53 to actually to go up. And in this case, they will be shifted towards the right-hand side of this graph. And for the rest of the genome, I would say, or the coding, for the rest of the genes that we are about to study, we would expect the vast majority of them would actually cause no effect to the p53 stability, and as such, they will likely be centered in the middle of the entire population. Conversely, if something actually whose loss will result in the p53 loss of stability, then they will be concentrated towards the left-hand side of this graph. So having said that, we actually performed our screens, and then we observed some result. This is the result, what the result will look like. And then I cluster those the effects on each genes across different mutants, and then I can actually test what are the behavior based on the behavior of those genes in each different mutants. Then I can cluster this mutant. I can functionally study them as a cohort. If we were to actually zoom in those results and see what are the top hits and from this study, it's actually pretty heartbreaking in the sense that a vast majority of the top hits is actually what we have already known to date. This is not a surprise to us because p53 has been studied for more than 40 years. But what is actually interesting is that this result actually validated our system of study, because we actually found all those ones that are actually known to regulate p53s. So interestingly, there are actually some genes that are actually still unknown to actually cause this p53 regulations, and this one naturally becomes our follow-up for the subsequent studies. So In this case, I just want to mention two of the top examples, which is FOX42 and CCDC6. The name is so kind of a mouthful of words. It's because nobody knows what these two genes actually does, and let alone their functions on PP3 stabilities. So as shown in this plot, I actually should explain that the blue color indicates that the loss of this gene will cause its PP3 to be destabilized, and red color indicate that the loss of this gene on the x-axis will cause this gene to be stabilized. So this is kind of the premise of our study. Now let's moving on to see what we can actually do with this data. So since this is kind of with a little genetic focus of our presentation today, I want to actually discuss briefly what we have done after this. So for this, since they are not known, then I moved to the a database called the cancer dependency maps. So basically, based on the behavior of losing a particular gene on cell fitness across 800 different cell lines that has been tested. And we generate this database that can actually help us to relate the functions of two completely unrelated genes. If two genes actually whose loss result in very similar phenotypes or fitness across the entire 800 cell lines being tested, then very likely these two genes are belonging to the same pathway, or they actually share or share within the same complex. So based on this, the premise, we actually looked into what is FOX and CCDC6 factors into the systems. Indeed, CCDC6 and FOX42 are the two genes that are actually having the strongest correlation throughout the entire data sets. As such, we suspect they belong to the very same genetic complex or same pathway. Then the question is, who is in this pathway then? So as we know from the genetics class again, so if gene A actually depends on my favorite gene of interest to achieve a goal, which is PP3 stabilities, then if I were to remove this gene of interest, then the effect of A on the PP3 stability would not be observed anymore. In this case, we call the gene A depends on or requires my favorite gene of interest to achieve this phenotype. Based on this very premise, I conduct the studies as follow. Since I've already performed the screenings, as we just talked about on the stabilities, what if I just perform the exact same screenings, but in this case, with the absence of CCDC6 or FOX42? In this case, I will be able to find out the genes that actually depends on the CCDC and FOX to regulate PP3 stability. In other words, I can find out who is in this pathway. As we can see on the left-hand side is actually deleting the FOX, and on the right-hand side is deleting the CCDC6, and I plotted this as shown. And as we know that for most cases, like for most genes, the removal of both would have the effect would not really change whether it was the presence or absence of CCDC or FOX, because we don't expect them to be in the same complex or pathways of these genes. What becomes interesting, though, is those that actually got knocked off this diagonal axis, whereas they do not have the same effect anymore after losing the CCDC or FOX. And this will naturally becomes our interest. One of the top hits in this case would become the USP28, as you can see here, who are being knocked off the axis, the diagonal here. In other words, they were a potent regulator of the PP3 stability when the CCDC and FOX was present, and they are no longer effective when these two genes were lost. Since USP28 is actually a well-known PP3 regulators, we now then suspect that FOX and CCDC regulate PP3 through USP28. A very similar kind of approach can also be done to find out what are the interactors of USP28 that are disappearing once we actually loses the CCDC or FOX. So the very there's only a few more slides left, and so the very last kind of idea is that so this is good, we have all that. So what is the functional impact then? As we already mentioned that if we stabilize the PP3s, mutant PP3s, then this causes the metastasis and tumor formation. The question is that what if we actually remove the two genes I just mentioned, which will ideally cause the PP3 to actually be destabilized? And will that actually causes reduce the potential to causes metastasis? This is indeed done in using mice to see that we use something called a metastasis assay, whereas we inject the cells that has been tagged with a green color into the tail of the mouse, and through circulations, the tumors will, the cancer cells will start to migrate into the lungs. And if they are actually cancerous or has a strong metastatic potential, they will start to form colonies inside of the lungs. If we compare the control group versus the group that actually we have removed CCD3 box, namely p53 now down, we would see a differential between the colonies that are being formed inside of the lungs. And in other words, the control group should see a huge amount of colonies inside of lung, whereas the ones with lower p53, they should see fewer. This is exactly what actually we have seen. So in the control group, here we were able to see that they have roughly about 400-ish average on average across 10 different lungs. We have 10 different mouse we have tested. Upon removal of either of the genes, and then we have about 200 versus the 400 colonies formed in this case. Thereby, this thing has been functionally checked out. As I mentioned, this is not the only gene that we have actually focused on. On the other side, if we remove this gene, this causes stabilization of the p53 as well, and those also are worthy follow-up. And indeed, we also follow up in a very different, approaches. And finally, I just want to kind of summarize what I've been saying here. So we know that p53 stability really matters. And the question is, we want to find out what are the ways that triggers the stabilizations. And to do that, we actually uses a CRISPR-based, reporter-based CRISPR genome-wide screens to do that. And in the real research, we actually intercept multiple omics approaches together, including proteomics studies, and also mining the public data sets. And all this helped us to actually hone into a few genes or pathways that are to be particularly focused on. And as I mentioned before, that all of this is subject to validations, biological validation, as we just mentioned. So all this will be subject to in vivo validations in mouse lines, and also compared to the human data towards the very end. So taken together, we have devised a genome-wide CRISPR screen tools to study the p53 stabilities, and we identified a pathway that is potentially regulating the p53s. And what's the next step? We are in collaboration with our Israeli colleagues in the Weizmann Institute. We are now we're now conducting drugs that are screening for drugs that can actually target this pathway and causes the reversal of the tumors via the destabilization of mutant p53s. So folks, if you're really interested in this project and also similar CRISPR technologies, please check out the JLSE summer research opportunities, and also many scholarships available through the Lunenfeld and SickKids, and also the HMD research courses as listed here. So thank you very much. And all of this would not have been possible with my lovely colleagues throughout in the lab and across the world, and also all the funding supports that has been provided to us, including the human biology and the new college scholarships here. So thank you very much for your attention, and thank you.
Thank you so much, Lou. That was so well done. Your slides really made something very complicated understandable, even to an audience that isn't your students. So I know a lot of your students are here. I'm going to turn it over to Colleen now, and ask Colleen to facilitate some questions.
Sure, I absolutely will do that. I have a couple main questions of my own, but I think we'll see what other people ask first. Najin, I think you might still be here. She's also with Human Bio and might have some things that she'd like to say. Is there anyone who has any questions for Lou? Bear in mind, you don't have to have a Human Bio background. You can ask more broadly about the general applications of this in the real world, and anything along the cancer line and its applicability. Does anyone have a question? I guess I need to be able to see the whole gallery. Hold on.
Francis has a question.
Francis, please go ahead.
Okay. I haven't taken biology since high school. And so,
Nice.
so
Okay.
I just want to, can I just check my like very rudimentary understanding? So P53 is a gene that is cancer protective, cancer suppressing normally, but sometimes it gets mutated and turns into mutant P53, and that's bad, and that helps cancer grow, right? And so you're trying to find what causes that mutation, and you're trying to like target the other genes that maybe make that mutation happen so that you can stop that mutation from happening. Is that correct? I'm sorry that I know you just explained that, but
Oh, no, not at all. Yeah,
my question then is like, why don't you just like zap out the mutant P53 itself instead of like, is it because you want to restore it to its healthy state so that it continues its like protective activity?
right. Thank you very much for the question. So I guess I'll answer this in two prompts. So the first is that indeed, so it took us actually almost 20 years to figure out that the relationship that PP3 is actually a tumor suppressor, but yet the mutant will actually cause us cancer. The question is that this is a known fact. This is happened already. So the PP3 is already mutated inside of our cells or inside of our bodies. The question is that the mutated PP3 doesn't really do much to harm us. It's the accumulation of the mutant PP3 that actually drives or causes the cancer. So our goal is to find out what can stop the accumulation of the PP3. So that's really what our focus is in this case. And in terms of why don't we actually just remove the mutant PP3 completely, the genes completely out completely. And so this is definitely an approach. If we can actually achieve very targeted, very precise editing of the genome, which is unfortunately not happening yet at this point. I think around the world, the only person who did it is a Chinese scientist who is now in jail and by editing the embryos. And it's just because, like, I guess it doesn't pass as the ethical concerns as well. But the question is that mostly is because we cannot yet achieve this very precise editing of that one base pair to reverse it back yet. So instead, since we are talking about a regulatory network, that gives us a more avenue to actually to target, and actually the effect is also amplified by targeting the regulatory effect. So I hope that kind of explains a little bit.
Yeah, thank you.
Scenario.
And Lou,
Yeah.
I'm going to check into, because this is not my field either, but when you say the accumulation of P53 is the problem, you mean P53 is really important for maintaining the cell cycle. And so when you say accumulation, do you mean the accumulation of cells because of the P53? Yeah.
Right.
So when the P53 is accumulating, it's literally because it's allowing the cells to continue to proliferate rather than stop. Correct? It's because of the tumor mass.
Yes. So I guess the question is that like, what is the mechanism of mutant P53 causes cancer? So I would say I guess the field kind of consensus in this case is that the mutant P53 alone is a lousy kind of a driver for cancer, and it only becomes effective once you actually aggregate them together. When the P53 actually start to accumulate in mass, then they were able to actually carry out the downstream functions. And our goal is to stop this process from happening. So you can exist on your own, and this is effect already happened. So, but all we want to do is to stop this aggregation from happening, and thereby preventing the cancer from forming. So in other words, what are the accumulation? There are accumulation of the mutant P53 proteins. So we want to prevent this step from happening.
Thank you. Thank you. Does anyone else have any questions?
I guess Tara summarized it very well.
Yeah, okay, Tara, go ahead.
I think she's
Tara,
muted.
Tara, you're muted.
Sorry, Lou, are you done your PhD thesis? Is there still some work to be done, or are the results that you showed us today enough for you to move forward? That's what my question is. What's the next step for you?
Oh yeah, sure. Thank you. So I guess one thing is because science drives us to there's never an ending to to a project. There's always things that we can drive forward, and such as I mentioned, like that we could divide using this reporter to devise drugs that can actually treat it to target this process. So I actually just carried out a study last summer in Israel, and we're still doing this now. And in terms of like graduations, that's negotiable with department, my department. And but then again, yeah, in terms of like to drive a thesis chapters, definitely this we could conclude at this point a particular chapter to in terms of the thesis is concerned. And
Okay.
so our
This
study,
is one study amongst many that you're going to be doing for your PhD, is that right?
yes, indeed. So we screening is only part of our branches. We actually perform multiple parallel screenings. Just now, like I mentioned, like so they have actually would be of interest to many of our audience that those are many of the functional genomics approach that can actually achieve this goal, as such as the ones listed here. Those are just many of the approaches here. So we actually performed all of them, and so it's actually the integration of those approaches together that actually allows us to hone in into several really hot hits to follow.
Great. And Lou, just to follow up, what do you want to do now that you have all of these skills? What do you want to do to continue using these skills after you graduate?
Right. Well, thank you very much. So I guess there are two things I want to mention. So first, this is a CRISPR is will very soon become a very, I guess, fundamental technique that everybody is such a useful arsenal to have to collect. And so one of my goal is to actually integrate this into our teaching labs. And in fact, in the I guess part of the 311 labs, it was 19 last year, we actually integrated some of the screenings and also the analysis of the public database as part of the courses. So that's one thing that we're hoping to bring this into my kind of teaching passions in one, and also to kind of wearing the research hat. We're hoping to actually further expand this tools or these pipelines to study potentially any proteins that we're interested in for its stabilities. And indeed, we are actually carrying out a collaboration already with a group while I'm here during the conference. We're already starting a discussion of the collaboration for a particular proteins using the exact same approach. So this will become, I would say, my future kind of research goals as well. So both the teaching and research will implement what I've learned from PhD. It's really the skills that obtained from the study that will be useful to propel my future career.
Brilliant, brilliant. Colleen, I think Francis has a.
Oh my gosh, Tara, you're so much better at this than I am. I can't see the hands in the corner. Of course they are. I apologize. Francis, go ahead.
I just want to say that, Lou, I think you will be an amazing teacher and researcher, because the sort of excitement and passion that you bring to describing your research is really infectious. Even though I can't totally understand it, I feel that it's really exciting.
Thank you very much.
The other thing I was going to say, though, is a question is like, just again, like I'm very ignorant of how things work in the sciences. So this is obviously like a huge project, and many other people are working on what you've described. So how does it work in the sciences when, like, at a certain point after like a kind of arbitrary number of years relative to your research, you have to be done and write a dissertation and graduate, but your research is not done. It must be ongoing for like years. So what do you write?
Right, indeed. So I guess there are two things that we'll also mention. So similar to a research grant, there's so much things we want to actually explore, but at a certain point that we really need to wrap up the story and let this field know what's going on and to let it publish. So this is actually an approach that we actually took. So we, after conducting like instead of following all the possible hits we actually found, we focused on one or two of them together and then derived our story as a kind of a confirmation of our strategy. But the next step is to actually follow up more in detail the mechanism and with drugs that are actually doing the job, can targeting it. So this will be our second or even the third papers in this case. So as mentioned, as Francis mentioned about the collaborations, so nothing can be done without a tight collaborations with scientists, not just within Canada or Toronto, is around the world. It's everybody contributing to this. Nobody can do science just on his or her own. It's the entire community that actually does it. And the result will eventually also benefit the entire scientific communities in large. So in terms of like when I should actually wrap up my thesis. And so this part is actually my chapter 1, what I just presented. So yeah.
Interesting. Such an interesting difference between the sciences and the humanities, because of course in the humanities, our research is also collaborative, but we kind of pretend that it isn't. Or I think we sort of force ourselves into working in these solitary ways that are sort of inauthentic to the reality. Sorry, go ahead.
No. And I just have one last question for Lou is like the P53 mutation is really common in cancers, but I can't remember which cancers this might be most relevant. Would this be relevant for every kind of cancer, or specifically are you targeting a certain type of cancer?
Yes, so that's a great question. So if we were to write a grant, definitely will say this is the, well, I mean, I should honestly say this is the most commonly mutated genes in all cancers, And of course, there are exceptions. Certain cancers actually, some really weird pancreatic cancers do not, a subtype of pancreatic cancers do not. And if we were, again, this is something that I think we brought into the 311 genetic labs. So we look at from the database, we can actually see what it's called the human cancer genome atlas and TCGA data sets. If we go in there, we can actually see what the mutations across different cancer type, even subtypes, and what are the distributions. So with some particular mutation, cancer types such as ovarian cancers, actually amounting more than 80% of the patients, and 80 to 90% of patients would carry mutations in PP3. And but the other extreme is those mentioned, like those really weird pancreatic cancers, which actually have near zero, that nobody actually, or neuroendocrine tumors that actually doesn't have any PP3 mutations, which is actually puzzling. Why? But then we realize it's actually downstream of PP3. It's their pathway actually got messed up. So that's where the cool thing is. And the other extreme is actually also something called a low-grade gliomas in the brain tumor, actually where the tragic hip singer actually passed away because of. And in that particular one, especially that particular cancer, so almost 100% of the cases actually carry the mutations in this PP3, and it's of a particular PP3 mutation codon. So that's
so really this has broad applications for almost every type of cancer.
I would hold on to that excitement, but I would say yes, affects a bunch of a variety of cancers. Yeah.
Thank you so much, Lou. That was an excellent job. Thank you very much for doing that. We're very
Thank you very much.
to have you. We're very happy to have you as our fellow this year. Thank you.
Thank you very much. And I guess the last public announcement is that if you're interested in the thing, our work, please check out our laboratory, Daniel Schratnick's lab, and also the possible research course that are associated with it. So we're definitely welcoming folks to conduct some research.
Thank you so much, Lou. So exciting to know here at New College, our students have access to this kind of cutting edge research. Really, really fabulous. And for the human biology students who are here, lucky you if you had a chance to work with Lou, and if you are able to learn from Lou and others around this research, I think it's an incredible opportunity. It's exactly 2:15, and we are going to move to a presentation by our colleague Amber, and Amber is from Buddhism, Psychology, and Mental Health, and I'm going to ask our program director, Francis Garrett, to introduce Amber.
Yeah, thanks so much. I'm really happy to do that. Amber Moore is a PhD student in the field of Buddhist studies in the Department for the Study of Religion, which is also my department. And Amber has been with us for a number of years studying intersections of Buddhist narrative literature that talk about visionary landscapes, for example, and also Buddhist ritual. She has a BA in Buddhism and philosophy and Himalayan languages from Kathmandu University in Nepal, and then came to us after that. She also has lived for many years in Tibet and Nepal with her family, and she works in many languages in Nepali and Sanskrit, Tibetan, and Newar to do her research that we're going to hear about today. And finally, I also happen to know that Amber is an incredible dancer and a ballroom dancer in particular currently. So that's been a real joy for me to see her performances on Instagram recently in particular. So Amber, please tell us about your work and where things stand for you right now.
Thank you so much, Dr. Garrett. Yeah, it's great to see you. And thank you, Tara, and everyone at the new college. It's been a real honor and pleasure to be associated with the new college this year with the Senior Doctoral Fellowship. And I really appreciated the support and all of the great events. Thank you so much, Lou. I really also appreciated your passion for your research. I can say that I also join in that with my own area, and I know how hard it is to stop and to write those chapters and to communicate what's been done before going on to the next area of research. So today I'm going to talk about my research, but I'm also going to do a bit of a meta reflection and invite you to be involved in that reflection on one of my chapters, my second chapter in particular, from my thesis. And I'm looking at basically how translation, now Dr. Garrett was saying that I work with various different languages, the Nepal Bhasa, so the indigenous vernacular language of the Newar peoples of the Kathmandu Valley, and Sanskrit in this particular instance. So it's not really involved with the Tibetan language, this project, but just to look at what my own translation is kind of doing and encouraging a bit of reflection. So I had 4 main questions that I wanted us to keep in mind. So I'm going to go over those questions first, and then I'm going to just do a bit of reading from my translation of this one narrative. And then hopefully we don't have that long, so we'll see how much we get through. And then we'll just invite some discussion. So I'll just go ahead and start with that. So today I'll be discussing recent research and translations in the field of Buddhist studies on the Buddhist narrative entitled the Manishaila Mahavadana, which includes an origin story of the Buddhist goddess Vajrayogini at the town of Sanku in Nepal. This is a multilingual narrative comprised of both popular, localized, and lesser-known Buddhist narratives called Mahatmyas and Avadanas. We will look at the problem of language in what is essentially a cross-cultural study due to the source and target languages originating from disparate cultures. Lydia Leo discusses the idea of both cultural and textual translation as an epistemological crossing, because when we discuss translations, we're not only talking about transferring from one language to another, but in terms of the ideas of transferring the ideas from one culture to another. And this is the sense in which I'm discussing the idea that translation is inevitably, and for better or worse, always going to be a form of commentary. And why is this? This is because the agency of the translator and the context in which the translation takes place is something that cannot and should not be ignored. And this is what I'm kind of reflecting on today. Okay, so I'll talk about 2 different layers. There's 2 levels of cultural or layers of cultural and linguistic translation in the case of the text that I am working on, the Manisharla Mahavadana. So I have two, just to give you some visual on that, because I don't have a PowerPoint today. We have this one text here. This is the multilingual Nepal Bhasa and Sanskrit version of the Manisharla Mahavadana. And it's a 80-page document here, and it includes a compilation of a number of different Buddhist narratives that have all been written in a way so that it's localized at one particular site in one town in Nepal. Then we have another manuscript witness here. This is a published book. The manuscript witness, I have just a copy of it here. So I have this. These are the kinds of things that I'm working with at my kitchen table. So we have this document here, which is a 40-page document of a fragmentary kind of manuscript scrawled in Sanskrit handwriting. So this is altogether we have 11 manuscript witnesses for this document, and or rather less manuscript witnesses and a few published works. So back to the two levels. The first case of cultural and linguistic translation or interpretation in the case of the text that I'm working on is directly apparent in the reading of my multilingual source text, which is the translation that occurs from Sanskrit to Nepal Bhasa. In this text here, this is what we have. So this text itself includes a translation of the Sanskrit for the Newar speaking demographic. Now I want to mention that Sanskrit itself is seen by Newar Buddhists as somewhat of a language of authorization. It's not a local vernacular in most cases, although Sanskrit is still spoken among some families. It's more of an elite language, very much a language that is reserved for the use and deployment by a select group of Newar Buddhist priests and initiates. The translation of the Sanskrit narrative into a more local vernacular that would be understandable to the Newar public would therefore be a move that would both alter the use of the text and will alter the audience of the text. It also requires a good deal of commentary in the process. So this is a process that was occurring in the 1950s and 60s in the family of Bhanavajra Bhattacharya and his elder brother, who were undertaking this project of compiling these local stories and Sanskrit narratives and translating them into the Nepal Bhasa. So it's kind of a move that really alters then the context and the use of the story. So in that sense, it's a literal translation. It also comes into a cultural translation because it changes the cultural use and application of the text. In addition to the explanation of the text, many choices had to be made in that scenario in terms of which vocabulary to use, how to rephrase the narrative into Nepal Bhasa from Sanskrit. And a lot of what my work on these texts does is look at the divergences on a very minuscule level, and the title names and the divergences between the Nepal Bhasa and the Sanskrit in the manuscript witnesses. And I look at what kind of erasure or inclusion is going on in that instance, and what kind of, and maybe look into why that was happening. But the person I understand who first opened up this literary version again, I just wanted to mention his name was Bhanavajra Bhattacharya, who is now passed away. Luckily, I was able to have an interview with his son, which was very informative and which will comprise another chapter of my thesis. But so now on to the next level. So there's the one level of textual interpretation that I'm looking at, but inevitably, the next level of agency and translation as commentary I'm looking at is in my own PhD research because it's founded on my study and my own translations of this same Buddhist narrative, traditionally read publicly or privately among Newar Buddhists on various special occasions. The purpose of the reading of this text in the context of Newar Buddhism is for the accumulation of merit, the attainment of liberation in Buddhist terms, for the ascension to the Pure Land of the Buddha Amitabha. And this is a narrative that has been preserved and passed on in Newar families for generations, as I've mentioned. In the process of translation, through my own choice of English grammar and terms, through choices of whether to adapt the text to English-speaking conventions or to try to stay closer to the originals, there is an epistemological reformulation that happens in the text, which goes beyond just translation from source to target language. As a translator, I feel it is imperative to reflect on my own agency, my own influence, the cases of erasure and expansion that also go on in my own translation into English, and also with regard to not only the language, but the cultural context then which my own translations would or could be used by in both the academic and the Buddhist communities. So I've taken lots of inspiration from Professor Lydia Leo in this for this talk. So she's come up with a number of valuable questions. I'd just like to introduce you to some of her work. Professor Leo, her research centers on modern China, cross-cultural exchange, and global transformation. She's a professor at University of Columbia. She focuses on the movement of words, theories, the evolution of writing, and textuality. In 2021, Professor Leo, she joined the ranks of the International Council for Philosophy and Human Sciences with UNESCO. Her publications are The Freudian Robot, Tokens of Exchange, The Problem of Translation in Global Circulation, Translingual Practice, which I will be quoting from today, Literature, National Culture, and Translated Modernity. And she also wrote The Birth of Chinese Feminism, Essential Texts in Transnational Feminism. So since the translation of Buddhist texts from Sanskrit to Nepal Bhasa to English is a project of cultural translation, and this must be recognized, it involves more than just the translation of one language into the target language. It moves resources between varying communities, and in this case, moves resources from a rare Buddhist text held by an elite Buddhist community to one that is wide-ranging and academic. As I see it, her questions fall into 4 categories surrounding the problems of language in textual and cultural translation. So first she asks, number 1, what can we say about translation and its implications for cross-cultural studies? We should ask in whose terms and for which linguistic constituency and in the name of what kinds of knowledge or intellectual authority does one perform acts of translation between cultures? This is a question that Leo feels becomes more acute, especially when there's a crossing from west to east or east to west. She quotes Leach when he states that linguists have shown us that all translation is difficult and that perfect translation is usually impossible. This brings us back to my point, which is the agency and influence that the translator exerts, which comprises a certain level of commentary that is often not recognized. The second category of questions inspired by Leo involves tropes of linguistic or cultural equivalence in translation. Leo reminds us of a famous quote by Borges, that the dictionary is based on a hypothesis, obviously an unproven one, that languages are even made up of equivalent synonyms. The third category, which is reminiscent of Assad's approach, is to always look at the dynamics or shifts in power. I might ask, how does my translation and my particular interpretation format, audience, facilitate certain shifts in power, shifts in institutional praxis, and either confront or endorse certain forms of authority and intellectual authority. We might ask, how will a translation be used, for instance, and by whom? How is the source language included or not in the text of the translation? And how can I adapt my translation by adapting to the needs of various translingual audiences? And I also wanted to add a note at the end about translingual practice in pedagogy as well, because I came across a few really helpful points about how to highlight students' own linguistic and cultural resources in the classroom. So that's something that came out of looking at this as well today that I'll get back to. So the fourth category of questions that Leo reminded me of was, how do we address and respond to what we feel are mistaken notions of corrupt versus classic language usage? And this is always coming up in my practice of translation, because my texts, they are written in Sanskrit very often, but Nepalese Buddhist Sanskrit is a very unique kind of expression of Buddhist Sanskrit and Sanskrit. It's not a classical form, and it's been seen very often as a more corrupted form. And in general, sometimes Newar Buddhism is seen as a corrupted tradition that's incorporated all kinds of Brahmanical views in modernity, and the language that's used is not classical Paninian grammar, and so a lot of issues come up with the text that I work on. And so what I'm working on is how to kind of see the value, see the uniqueness in this kind of divergent form of Sanskrit. And we see the kind of patterns that come about with the use of Nepalese Buddhist Sanskrit. Tuladar Douglas has written quite a bit about the grammatical details of how you can kind of track that. I've noted a lot of vocabulary that's often used in the Nepalese Buddhist Sanskrit. So it's coming together as more of a unique form of Sanskrit itself, not just that it's a corruption. And the fact is that if I ever amended it, or if you have an emendation of a Nepalese Buddhist Sanskrit text, it will then become completely unusable by the Nepalese Buddhist priest or in that lineage, because Nepalese Buddhist texts, as they're written, whether it's a stotra, a song, or a narrative, in the form in which they are written, they're completely understandable and completely usable in that form, because what we often see is a slight mixture of the Nepal Bhasa, of the vernacular that comes in the Sanskrit, and a slight erasure that happens of the Sanskrit grammar. But if you are having a translingual kind of approach to this form of Sanskrit, then it becomes completely comprehensible in the form that it's in. So I find that this is kind of a really good model just across the board. And if we were to apply it to English in our own classroom, just to kind of ask ourselves, what would that happen? What if we see the unorthodox use of English in our own classroom by people who are multilingual, for example, as not necessarily incorrect, but just an unconventional or a unique approach? Okay, so anyway, well, while in certain instances one needs to distinguish proper usage of language, on the other hand, we might see some atypical use of Sanskrit or of English, and this is a practice that could be addressed in a sensitive manner by not jumping to the conclusion that Nepalese Buddhist Sanskrit, for instance, is corrupted or mistaken. This is an approach that can also be encouraged and explored in the classroom, which I mentioned. Instead of using the term corrupted or incorrect, I often refer to Nepalese Buddhist Sanskrit that I work with as an unorthodox grammatical usage of the language. And in my reading of Nepalese Buddhist texts, the many song stotras, it's a good, provide many good examples across that are kind of uniform across the board. And then I mentioned the emendations are not especially welcome within a certain scholarly demographic, while they would only be helpful to a very elite group of classical Sanskrit academics. And so keeping those, all of those things in mind, now what I wanted to do is just maybe have a little bit of fun actually reading through a translation. So now I'll share my screen, and let's see, sharing screen from, I think it's the second chapter. Okay, so is that all good? Tara, okay, thank you. Yeah, so you can see a little bit about what I work on. So we have a comparison here of the chapter titles throughout the variant languages, and focus on what kind of terms were left out. So we come here to the structural comparison of first and second chapters from the Manishala B, which is the text at hand, and the fragmentary. So we can see we have the English translation paired against the Nepal Bhasa, which also includes Sanskrit. And then we have the actual, here on the far right, we have the actual Sanskrit text, which is which includes very, very little. It's just a few select passages are there present in the Sanskrit text, where it's much more expanded upon in the Nepal Bhasa. So you can see here how it's kind of been compared. You can see what's present in the Sanskrit text over here. Then you have the equivalent in the Nepal Bhasa here, that's bolded in the middle. And then you have the same thing over here in the English, so we can see how the three texts compare across the board. So I just wanted to skip to page 48, and I'll read from from chapter 2, so we can actually get a sense. Okay, so I believe this is the tale about the emergence of Vajrayogini and some of the experiences of her disciples after they had a vision of her on the mount, on the Manishala mountain. So this is chapter 2. Once again, Maitri Bodhisattva beseeched the glorious Lord, O Bhagavan, in the area of which Tathagata was the glorious goddess Ugratara Vajrayogini, in the form of a divine flame covered and then ritually established as a divine image, Murti. The wish to listen to this story having arisen, Maitri Bodhisattva requested, "Please do tell." The glorious Lord replied, "O Maitri Bodhisattva, I will tell you this story, so listen and rejoice." How did the story go? Sometime after the glorious Sikhi Tathagata had passed into nirvana during the Bhadra Kalpa, the lifespan of beings had reached approximately 60,000 years. At that time, in the city called Anupama, the Tathagata called Vishvabhu was born. At that time, I was known as the Bodhisattva Parvata, who was serving Vishvabhu Tathagata. At that time, the glorious goddess Ugratara Vajrayogini arose as a light and took on a distinct form. How is the one in the form called the triple image, Trimurti, described? She had 3 faces. Her main face was red, her right face was blue, and her left face was white. Each face had 3 eyes. She held aloft a sword in her right hand, and she held aloft a blue lotus in her left hand. She appeared as a wondrous and beautiful youth of 12 years of age, smiling sweetly, and she was replete with all auspicious marks and signs. In every way, the goddess was most excellent. Among all, she is most fit for worship and offering. She is the one who grants all of the various results for those who make aspiration prayers, establishing all the gods, demigods, and human beings in the state of bliss. She is said to grant the fruition of liberation. She possesses the triple nature, comprised of the qualities of pristine being, grasping conceptuality, and ignorant inertia. She manifested, taking on her universal form. As soon as the glorious goddess Uttara Vajrayogini had manifested, having arisen, the entire earth trembled. The divine beings sent down a shower of flowers. They played drums and musical instruments and exclaimed auspicious words. In the 10 directions, there was happiness, and all of the beings of the world were pleased. To the left of the side of the glorious goddess Uttara Vajrayogini, one Chaita called Mani Chaita also emerged in the form of a Mani stone, Mani Shila. This king of Chaitas would become renowned as Yogeshvara Chaita in the Kali Yuga era. Then, facing the front of the glorious goddess Uttara Vajrayogini, Akshobhya Tathagata appeared in the shape of the Naga Vasuki in the form of a Mani stone, which arose spontaneously of its own accord. Then, about 28 yards below, to the right side of the glorious goddess Uttara Vajrayogini, there spontaneously appeared a triangular-shaped Mani stone, the manifestation of the local protector, the glorious Mahakala. Immediately after that Mani stone appeared, Mahadeva Narayana, the gods, demigods, human beings, yakshas, rakshasas, kinnaras, and maharagas all arrived at that place to worship her and make offerings. In this way, they remained in service to the glorious goddess Uttara Vajrayogini. At that time, a person called Yoga Yanacharya was performing his duties as an acharya. Starting out very early in the morning, before sunrise, he would bathe and perform a puja for the glorious goddess Uttara Vajrayogini. Then he would sit in the cave called the Cave of Accomplishment, where he practiced the daily meditation and recitation of the root mantra of the glorious goddess Uttara Vajrayogini. And in this way, he served her as a devotee 2 times a day. After Yoga Yanacharya had spent some time in this way, one day the glorious goddess Uttara Vajrayogini, being extremely pleased with the way he had meditated, appeared directly in front of him, just as he had been propitiating her. And she said, "O son Yoga Yanacharya, I have witnessed your feeling of devotion and am pleased. Whatever kind of boon you would like to request of me, please request it now." Thus spoke the glorious goddess Uttara Vajrayogini. Having heard these words directly from the glorious goddess Uttara Vajrayogini, Yoga Yanacharya was happy. Seeing the glorious goddess Uttara Vajrayogini appearing in her own form, he was so amazed and overcome with devotion that he jumped up from his seat and circumambulated her 3 times. He bowed down at her lotus feet, and with his hands folded in Anjali, he responded, "O mother, I am not supplicating you in order to request a worldly boon of any kind, like this or that. Whatever it is that you see fit to grant to me, I will be satisfied with just that." Having beseeched, he remained silent for a moment. Hearing the words of Yoga Yanacharya, the glorious goddess Uttara Vajrayogini was delighted and replied, "O son, O sweet boy, I am so happy having heard your aspiration, and for this very reason, I know that you should be the one who maintains my daily Nitya Puja and so forth from now on. Furthermore, in the future, I would be pleased if your lineage would continue to undertake all the activities and duties related to worship. For as long as your lineage remains steadfast in this place, know that until then, I too will remain here at my place. But if a time comes when your lineage fades into decline, know that I will forsake this place. Know this." Thus spoke the glorious goddess Uttara Vajrayogini. Yoga Yanacharya was delighted to hear such an address directly from the glorious goddess Uttara Vajrayogini. Once again, he folded his hands in Anjali Mudra and praised her with the stotra. Homage to Mahamaya, who bestows yoga insight. The 5 elements are subsumed in you. Homage to you who are one with Brahma. I pay homage to you, the creator and maintainer of this world. Forever you abide as the nature of creative power within all beings. You are the strength that overcomes. You give life to that devoid of life. Since death is overcome by your power, I pay perpetual homage to you. I pay homage to you in the form of a divine flame, manifested and unmanifested. Your peaceful form, the divine fiery one, homage to your brilliant form, Lord of midnight and Lord of day. I pay homage to you with and without form forever. Freed from the passions, I pay homage to you who has crossed the ocean of afflictions. You are beginningless, not of this world. You are fully liberated from the world. I pay homage to your universal form, subtle form, and material form. You are present in the various activities throughout the triple world. I pay homage to you, Pragyaparamita, who bestows the wisdom of complete awakening, Bodhijnana. To you, the mother of all beings of the triple world, in her forms, I pay homage eternally. Okay, so I'll stop reading there. But if we were in a normal context in Nepal, the reading would go on and on for days, and it's considered to comprise a great accumulation of positive merit and virtue, which will then result in having so many good karmic fruitions, including being born in the Pure Land of Amitabha. So just to give you a sense of the kind of work that I'm doing, and also the kind of meta questions that I have about the translation and the cultural context in which it ends up in the academic setting. So maybe we'll open it up now for questions and for discussion. And I'll stop sharing the screen, but I can share again if we need to.
Thanks so much, Amber. It's really interesting to read the kind of translation work that you've been doing. To hear it. Do we have questions from anyone else first? I have a couple questions, but Tara?
Thank you so much, Amber. It's been a long time since I've had somebody read a story to me, so that was really a nice way to spend some time this afternoon. Could you tell us a little bit about what drew you to this work, both to the questions of translation and to this particular text? In starting your PhD, there were many places you could start. Why here, and why translation?
Well, it was kind of serendipitous in a way. I knew that I wanted to work on local narratives in Nepal because I was with my one supervisor from Wilfrid Laurier, and he was working on localized narratives of Avadanas in Gandhara. So over in the regions of Afghanistan and Pakistan, where there's no longer really a living Buddhist tradition of the transmission and public reading of these narratives. So in Nepal, you have sometimes even the same Avadanas or the same stories are localized in Nepal, but you will see are localized in Gandhara, and you have a living tradition. So that was Dr. Jason Nealis. He really inspired me to engage in the realm of these localized Avadanas. And I chose Sanku because of its historical significance for Buddhist studies. It's been pointed out by a number of really famous scholars, and it's kind of a new area that hasn't been explored yet. And I also visited Sanku when I was about 15. So I have wonderful memories of visiting the monastery there. And I remember that I made it to the top of this long staircase, and I remember meeting the Newar Buddhist priest there. And at that point, we weren't allowed in the monastery. The foreigners, Westerners, weren't allowed in the monastery. And so I made it to the top, and I just kind of spent some time there and went back down. I was a bit disappointed. But I thought it was interesting that I went there when I was so young. And then when I made it finally many years later to University of Toronto, my supervisor, Dr. Christoph Emmerich, said that somebody had accidentally sent them this narrative of Vajrayogini at Sanku in a box of books, a box of Newar books that they had sent to him for his collection. And so it was perfect because this is the narrative of Vajrayogini that's localized at Sanku in the area that I wanted to work in. And it just kind of appeared in that way. So I had the long time interest, and then this text appeared, and I said, okay, so I'm gonna look at this text. And it was only later that we then started to find some very rare Sanskrit manuscript witnesses for this same text. And originally we didn't know, and some other scholars who were working on it's like Bhagopal Shrestha from Nepal, we didn't know if there was a Sanskrit version. So a lot can be learned from then comparing these presumably older Sanskrit versions of this text to the newer, because we didn't know how much cultural translation, how much adaptation, how much liberty had been taken in this Nepal Bhasa translation, because it would have been for the broader demographic as a Nepal Bhasa speaking public. And I could see, interestingly, that a lot of it, a lot of this kind of what we would normally think of syncretic language, of the inclusion of these Brahmanical ideas and the idea of Vajrayogini, who's a Buddhist goddess, as kind of like this great goddess in a sense of the Shaiva and Brahmanical traditions, was carried out throughout all of the manuscript versions in Sanskrit, and that's not something that changed. Whereas something that was altered was the more esoteric Buddhist language, was what you see was the erasure that happened then throughout. So that's kind of like the trajectory from beginning to end of my thesis.
Thanks so much for that.
Michael, you have a question?
Hi, good to see you. Thanks for coming, Michael. I really appreciate it.
No, it's great. Hey, Amber. Hey, Francis. Hey, Tony. Oh, well, Laura. Yeah, so I really enjoyed that. Thanks for that auspicious narrative. I'm kind of wondering, Buddhists themselves have been engaged in translation for centuries. And I really enjoyed the way you talked about translation, the way Western theorists talk about it. But I'm wondering, can we bring this into conversation with maybe the way Buddhists in Asia have talked about language or translation? Have you been able to talk to any Newar priests about how they react to sort of hybrid Sanskrit incongruous terms, or just Buddhist ways of thinking about translation and language? And I mention it in part because my advisor, Jose Cabezon, has a book I really enjoyed on Buddhism and language, kind of talking about ways Buddhists think about language itself, which might be helpful. But yeah, what do you think?
Yeah, absolutely. And I think that the differences have been really striking. A lot of this has been a reflection on what I've learned basically about Buddhism as a scholar and about translation as a scholar, and basically how it works. So if we talk to like Raju Shakya, I mean, good friends of mine, Dr. Narashman Bhattacharya, it's basically taken for granted that anybody who is going to be sharing a narrative obviously will be qualified, will be a holder of the lineage, and will have the freedom to basically elaborate on the text to make it relatable to the audience. So it's assumed even if you're translating, it's for the purpose of communicating to a specific audience, and it's expected to adapt that narrative to that audience. So if you're a Buddhist priest, then you would be using a Sanskrit text basically as an outline. And we could see that that's what happens in Gandhara. We have basically these outlines that are then in writing that were then presumably expanded upon. And so I think sometimes I had an underlying assumption that first of all, yeah, the equivalency assumption that the translator is supposed to be invisible, and that there are these automatic linguistic equivalents that you will ideally aim for to have a more accurate translation. But I do see from the Nepalese side and from those who are both scholars and practitioners, it's a little bit, it seems less rigid than the academic side, which is more focused on accuracy and the translator not having an effect on the text. From the Nepalese side, it seems to be assumed. But you have to understand the different contexts. I'm obviously not a qualified Buddhist priest, so I almost wonder, so in what context will this story even be read in English? And then would it be used by the Buddhist community, maybe a Nepalese English-speaking community, or sometimes, for example, like Dr. Bhattacharya, he comes to the West and he gives Buddhist teachings in English. So then would he use my translation? And under what circumstances would he consider using my translation? Have I adapted it too much or not enough? Or would he then further adapt my own translation using it as a basis? Would it even be helpful for him at all? I'm not sure. And I have to appreciate the fact that, and I'm very accepting of the fact that in this kind of niche area of study, it's really a process of self-exploration and self-learning. I don't truly expect that my research will be utilized much at all by anybody. Because especially in Newar Buddhism, it's a very elite group. It's not as open to the public. It's not normally taught in English. A lot of the initiations and the rituals are not open to an English-speaking demographic, other than Nepalese or Newars, who are obviously like bilingual, or, so I don't know if that answers any of your questions, but yeah, thank you for emphasizing that. And I really want to, I really want to make it more clear in my research that I have been definitely probably more invested in that side of thing than the Western side of it, but sometimes it's not always obvious. Yeah. Hi, Anthony, are you raising your hand? Oh, I can't hear you though.
Oh, can you hear me now?
Yeah.
Well, wonderful to see your talk, and I think it's been maybe a couple of years since I've seen you give a talk, but I haven't had a chance to see you, so hello to you and to everybody. I really liked how you, oh, sorry, here, let me lower my hand there, getting excited. I love how you look at the relationship between pedagogy and translation in your research, and I think that's really important. And I know you've taught a few classes, so I guess one of my questions will be around that. I have two questions. So how do you use this idea of translation in your classes? And you were kind of saying like this maybe like non-normative style of English. How would you bake that into some of your assignments to open up Buddhist studies to different people? Because I know sometimes like Buddhist studies has a certain kind of, it's a writing heavy set of courses usually. So a lot of students who might otherwise be attracted would avoid it for those reasons. So I guess I'm wondering, how does your approach on translation feed into your pedagogical approach? And then my second question is your kind of style or your approach to translation, how does it maybe challenge some of the kind of dominative narratives that, or some of the kind of methodologies that we see in Buddhist studies? And so I guess my question is like, how are you trying to maybe change the culture of Buddhist studies? And what would your kind of research do in your approach to translation, not looking at this kind of Sanskrit as a degraded form, but looking at it as, a Nepalese or form or Newar form, or maybe a vernacularization. I'm not sure if you'd want to use that term, but how are you maybe, like how would your kind of research